feeling totally wiped out after a busy day, or maybe you're buzzing with energy after a really good meal or a great workout. We all know these feelings, the slump, the surge, the constant push and pull of needing fuel and using it. But have you ever stopped to think about what's actually powering you?
Yeah, deep down.
It's far more intricate than just the food hitting your stomach. It's an incredibly sophisticated, ongoing chemical economy deep inside your cells, managing your body's ultimate energy currency.
That's the core of it, really. Everything you do, from the silent work of your organs while you sleep to the complex calculations happening in your brain right now, even the simple act of lifting a finger requires energy.
Right.
And your body has devised this, well, remarkable system for generating, storing, and precisely deploying that energy at the molecular level. It's the fundamental engine that drives all of life's processes.
And today, we're going to pull back the curtain on that engine room. We're taking a deep dive, guided by insights from a foundational text in human physiology.
A really solid source.
This isn't just a quick overview. This source provides the kind of detail that, once you understand it, gives you a profound grasp of how your body really works.
Yeah, it goes deep.
Our mission is to unpack this information, break it down, and go deep enough so you don't just learn about it, you truly understand it, like mastering the language of your own internal power system.
We'll be getting up close and personal with the key players in this energy economy. At the center is ATP, the body's universal, ready-to-spend energy currency.
ATP, okay.
We'll look at phosphocreatine, the crucial quick-access backup store.
Got it.
And we'll explore the different metabolic engines that extract energy from the foods you eat, the aerobic system that needs oxygen, and the anaerobic system that can work without it for a short time.
Okay, let's start at the absolute beginning then, with that central currency.
ATP, adenosine triphosphate. It sounds complex, but conceptually it's actually quite elegant.
Okay.
ATP is the molecule that serves as the direct, immediately usable energy source for almost every single function performed by your cells.
Like cash in your wallet, is that a fair analogy?
Exactly. That's a perfect way to think about it. If you need to buy something right now, you reach for cash. ATP is the cellular equivalent.
Right.
When a cell needs energy to synthesize a molecule, contract a muscle fiber, pump an ion across a membrane, or send a nerve signal, it spends ATP.
Okay. Now, the source also mentioned another molecule, GTP, guanosine triphosphate, playing a similar role. Is ATP the only currency then, or?
That's a good point. While ATP is overwhelmingly the primary energy currency for most cellular work, the source does note that GTP acts as a similar energy transferring nucleotide in certain specific processes.
Okay, specific.
Think of ATP as the US dollar, widely accepted everywhere in the cellular economy. And GTP might be more like, say, the euro, also a major currency, but maybe used in more specific transactions.
Ah, okay. So similar function, but maybe less universally used than ATP.
Precisely. Recognizing GTP's role adds another layer of sophistication. It shows energy transfer isn't strictly limited to just one molecule, even though ATP is undeniably the dominant player.
So this crucial ATP molecule doesn't just appear out of nowhere. Where does the body generate this currency? How do we make this cash?
Right, it has to come from somewhere. ATP is synthesized from the metabolic breakdown. The source uses the term combustion, which is vivid.
Yeah, like burning fuel?
Kind of, but it's really a highly controlled, multi-step series of biochemical reactions within the cell, and it uses the macronutrients you eat: carbohydrates, fats, and proteins.
Okay, so breaking down food. And these pathways are different depending on whether you're breaking down carbs, fats, or proteins.
Exactly. They follow distinct routes. For carbohydrates, primarily glucose, but also other sugars like fructose, there are two main stages or pathways.
Two main ones for carbs.
Okay. First, there's glycolysis. This is an anaerobic process, meaning it doesn't require oxygen.
Anaerobic, no oxygen.
Yeah. And it happens out in the cytoplasm, the main fluid-filled space of the cell, outside the specialized compartments like the mitochondria. Glycolysis breaks glucose down into smaller molecules, like pyruvic acid.
Okay, step one, glycolysis. What's the second route for carbs?
The second major route is the citric acid cycle, sometimes called the Krebs cycle. This one is aerobic.
Aerobic, so it needs oxygen.
Absolutely. And this takes place inside the mitochondria. Those structures often called the cell's powerhouses.
Right, because that's where the main energy production happens.
Exactly. That's where the bulk of aerobic energy production occurs. Pyruvic acid from glycolysis can enter the mitochondria and feed into this cycle if oxygen is present.
Okay, so carbs have glycolysis, then the citric acid cycle. What about fats? How do they get turned into ATP?
Fatty acids, which are the building blocks of fats, are broken down through a process called beta oxidation.
Beta oxidation, okay.
This is also an aerobic process, and it also happens inside the mitochondria. Beta oxidation essentially chops up the long fatty acid chains into smaller units that can then enter the aerobic pathways, feeding into the citric acid cycle, similar to what happens with carbs.
So fats also end up in the mitochondria, using oxygen. What about proteins? They're fuels too, right?
Yes, they can be used for energy, though the body often prefers carbs and fats if available. Proteins are first broken down hydrolyzed into their individual amino acids.
Okay.
These amino acids can then enter the energy pathways at various points. Many are converted into intermediates that fit right into the citric acid cycle. Others are degraded further into molecules like acetyl coenzyme A, which is a major entry point into the citric acid cycle.
So proteins can also fuel the aerobic engine.
Yes. Ultimately, the complete breakdown of amino acids for energy is also an aerobic process, eventually yielding carbon dioxide and water and generating ATP along the way.
So food goes in, carbs, fats, proteins gets broken down through these specific pathways, aerobic or anaerobic starting points, and the result is ATP. What makes ATP itself so powerful? It's something about those phosphate bonds, isn't it?
That's the absolute key. The ATP molecule, adenosine triphosphate, has 3 phosphate groups attached. The bonds linking the last 2 phosphate groups are often called high-energy phosphate bonds.
High-energy bonds.
Right. When the terminal, the very last phosphate group, is cleaved off by adding water, that's hydrolysis, it converts ATP to ADP, adenosine diphosphate, plus a free phosphate group. And this reaction releases a significant amount of usable energy, free energy.
And the source gave specific numbers for that energy release, didn't it?
It did, and it made a very important distinction. Under standard laboratory conditions, which are quite different from the inside of a living cell, think specific temperature, pH, concentrations.
Right, not real-world conditions.
Exactly. Under those standard conditions, breaking one of these high-energy bonds releases about 7,300 calories per mole. That's using the small c calorie, the gram calorie, or 7.3 calories per mole, with the capital C for kilocalories.
Okay, 7,300 standard. But the physiological conditions inside the body are different.
Crucially different. You've got body temperature, 37 degrees C, specific concentrations of ATP, ADP, and phosphate, a regulated pH. Under these actual physiological conditions found inside your cells, the energy released when that high-energy phosphate bond is broken is substantially higher.
How much higher?
As much as 12,000 calories per mole, maybe even a bit more under some circumstances.
Wow, 12,000. That's quite a jump from 7,300.
It is. And that 12,000 calories per mole figure is the one that truly matters for understanding how ATP powers processes in your body.
12,000 calories per mole. Is that energy sufficient then to do the work the cell needs, like building things or moving muscles?
Absolutely. The amount of energy liberated from breaking just one of these high-energy phosphate bonds in ATP is generally more than enough to drive almost any single chemical reaction step required in the body.
Provided it's coupled correctly.
Yes, provided the energy release is efficiently coupled to the reaction that needs the energy push.
Okay. And the source mentioned that if a reaction needs less energy, say only 500 calories, the rest of that 12,000 is lost as heat. That still feels a bit, I don't know, inefficient, like leaving change on the counter.
Yeah, from a pure energy capture viewpoint, maybe it looks that way. If a specific reaction only needs a small push, and you use this big 12,000 calorie packet, the surplus energy is indeed released as heat.
So just wasted heat.
Not entirely wasted. First, this large energy release helps ensure that the coupled reaction proceeds rapidly and essentially goes to completion. It really drives the process forward strongly.
Ah, okay. It overcomes any energy barriers easily.
Precisely. And second, as we'll definitely discuss later, that heat isn't just lost in a biological sense. It contributes significantly to maintaining your body temperature, which is critical for all other reactions to occur properly.
Right, thermoregulation.
Exactly. So while it might look like leftover energy from one specific reaction's perspective, it serves essential functions in driving reaction forward and keeping you warm. It's part of the system's design.
Okay, that makes more sense. So we understand how ATP is generated from food breakdown, and that it releases this powerful, physiologically relevant burst of around 12,000 calories per mole when its bonds are broken. Now let's dive into how the body spends this cellular cash. Where does this energy go to work?
Right. Making it is one thing, using it is another. The energy from ATP is the fuel for literally everything that requires an energy input in the cell. A major category is the synthesis of cellular components, basically, building the molecules that make up the cell and allow it to function.
Like making proteins, that must take a lot of energy.
Protein synthesis is a prime example, and yes, a major consumer of ATP. The formation of a peptide linkage, that's the bond that connects one amino acid to the next in a protein chain, requires energy input. Doesn't happen spontaneously.
And the source gave some really striking numbers here. It said the energy stored in that peptide linkage itself is relatively small, maybe 500 to 5,000 calories per mole, depending on the specific amino acids involved.
Right, not a huge amount stored in the bond itself.
But then it highlighted that 4 high-energy phosphate bonds from ATP are expended for every single peptide linkage formed.
Yeah.
4.
This is one of those details that really makes you pause and appreciate the body's strategies, isn't it? If we're getting roughly 12,000 calories per mole from each ATP bond under physiological conditions,
Right.
then expending 4 means the cell is investing approximately 48,000 calories.
48,000.
To create a bond that only stores, at most, 5,000 calories, that's nearly a tenfold difference, sometimes even more.
Why such a massive overspending? It seems hugely inefficient if you're just thinking about storing energy in that bond.
It's not primarily about maximizing energy storage in that specific bond. It's about ensuring the reaction happens, and happens efficiently and quickly.
Ah, driving the reaction.
Exactly. Chemical reactions proceed based on changes in free energy. Forming a peptide bond actually requires an input of energy. It's what chemists call an endergonic reaction. To make this energetically unfavorable reaction happen reliably and fast enough for life, the body couples it with the hydrolysis of ATP, which releases a large amount of energy in exergonic reaction.
So coupling an unfavorable reaction with a very favorable one.
Precisely. By using the energy from 4 ATP bonds, the total energy released, that 48,000 colmol, is so much greater than the energy required and stored, the 505,000 colmol, that the overall coupled reaction has a very large negative free energy change.
Making it highly likely to proceed.
Highly likely and rapidly. This huge energy push drives the reaction strongly forward, making protein synthesis occur effectively. The cell prioritizes getting the crucial job of building proteins done quickly and reliably over capturing every last bit of energy in the product bond itself.
That's a fantastic illustration of biological strategies spent big up front to guarantee the critical process completes without a hitch. What other synthesis tasks does ATP power?
Oh, quite a few. The source lists several others. For instance, the synthesis of glucose from simpler molecules like lactic acid. This happens, for example, in your liver after intense exercise when lactic acid builds up.
Rebuilding fuel.
Exactly. Also, the synthesis of fatty acids from acetyl coenzyme. This is part of how your body converts excess calories, maybe from carbs or protein, into stored fat.
Building fat stores.
Right. And the creation of other complex molecules like cholesterol, which is essential for membranes, and hormones, phospholipids, the main components of cell membranes themselves, and various hormones. All these building processes require ATP.
And then there was urea. The source specifically mentioned ATP is used for urea formation. That one seemed odd. Why spend energy making something the body just gets rid of as waste?
Yeah, that's another one of those counterintuitive points that highlights a critical biological need. Urea itself is indeed a waste product, mainly containing nitrogen, and it's excreted by the kidneys in urine.
Right.
But urea is the body's way of safely packaging and neutralizing ammonia. Ammonia is produced naturally when amino acids are broken down for energy or other purposes, and ammonia is highly toxic, especially to the brain and nervous system.
Ah, okay, so it's a detoxification process.
Exactly. The concentration of free ammonia in your body fluids must be kept extremely low. The process of converting this toxic ammonia into the much less toxic urea, which happens primarily in the liver, requires significant ATP input. So while urea is destined for excretion, spending that ATP is a vital investment in detoxification, preventing the buildup of a potentially deadly substance. It's essential energy spent on waste management and safety.
Okay, that makes perfect sense now. Energy spent to avoid poisoning yourself. Moving beyond building blocks and waste processing, where else does ATP power things? Big energy users.
Muscle contraction. This is one of the most energy-demanding activities your body performs, and it relies directly and immediately on ATP.
How does ATP power the actual movement?
Within the muscle fibers, there's a key protein called myosin. Myosin is one of the proteins that actually pulls on another protein, actin, to cause the shortening or contraction. And myosin also acts as an enzyme, specifically an ATPase.
ATPase meaning it breaks down ATP.
Precisely. Myosin binds to an ATP molecule, hydrolyzes it to ADP and phosphate, and the energy released from breaking that high-energy bond causes a change in the shape or conformation of the myosin molecule. This shape change is what allows it to pull on the actin filament, driving the sliding process that underlies muscle contraction.
And the energy demand for muscle can absolutely skyrocket, you mentioned earlier.
Dramatically so.
Yeah.
The source note that a resting muscle uses relatively little ATP, just enough for basic maintenance and readiness, but during short, maximal bursts of contraction, think lifting a very heavy weight or jumping as high as you can, the rate of ATP usage can increase by a staggering 150 times compared to the resting rate.
150 times. That's huge.
It is. This huge, rapid demand is a key challenge the body's energy system has to meet instantly. While the precise molecular dance of muscle contraction is detailed elsewhere, the crucial point here is that ATP is the essential and immediate energy source for making it happen.
Okay, muscle contraction, a huge ATP consumer. What about moving things across membranes, like getting nutrients into cells or pumping wastes out?
That's active transport, another major ATP consumer. This is when the cell needs to move substances against their natural direction of flow, against their electrochemical gradient.
Against the gradient.
Meaning pumping them from an area where they are at low concentration to an area where they are already at high concentration, or moving a charged ion towards an area that already has the same charge, or away from an area with the opposite charge. This requires energy because you're working against the natural tendency of substances to simply diffuse down their concentration gradient or move according to electrical potential.
Like pushing something uphill instead of letting it roll down.
Exactly. It takes work, it takes energy. The source gave examples like electrolytes, ions like sodium, potassium, calcium, glucose, amino acids, and even things like acetoacetate, a type of ketone body.
And where does this happen?
All over the body, across the membranes of virtually all cells to maintain their internal environment. Very importantly, in the kidney tubules, where your body needs to reclaim valuable substances like glucose and amino acids from the fluid that's being filtered out of the blood, preventing them from being lost in urine. And also in the lining of your gastrointestinal tract to absorb nutrients from your digested food into your bloodstream, even when the concentration of those nutrients might already be higher in the blood than in the gut.
So ATP powers the pumps.
It powers the specific protein pumps and transporters embedded in these membranes that perform this uphill work.
And glandular secretion, getting substances out of cells to be released, like hormones or digestive juices. Does that also need ATP?
Yes, often it does for a couple of reasons. The principle is often similar to active transport. Glandular secretion frequently involves concentrating the substance within the glandular cell before it's released.
Ah, okay, packing it up first.
Right. This concentration step might require moving the substance against a gradient inside the cell, which, again, is an active transport process powered by ATP. Additionally, the actual synthesis of the organic compounds that are being secreted, like protein-based hormones or digestive enzymes, also requires ATP, just like we discussed under cellular synthesis earlier. So both making the product and sometimes concentrating it for release can be ATP dependent.
Makes sense. Finally, the source mentioned powering nerve signals. How does ATP fit into something that seems primarily electrical?
Yeah, this one's fascinating because it's a bit indirect. The energy for actually transmitting a nerve impulse, the action potential itself, doesn't directly come from ATP breaking down at that exact moment.
Oh, where does it come from then?
The immediate energy for the nerve signal comes from potential energy that the cell has already stored up. It's stored in the form of carefully maintained concentration differences of ions across the nerve cell membrane. Primarily, there's a high concentration of potassium ions inside the nerve cell and a low concentration outside, while there's a high concentration of sodium ions outside and low concentration inside. These gradients represent stored electrical and chemical potential energy.
Like a charged battery.
Exactly like a charged battery. When the nerve fires, specific channels in the membrane open briefly, allowing these ions, sodium rushes in, potassium rushes out, to move rapidly down their respective gradients, driven by that stored potential energy. That rapid ion movement is the electrical signal, the action potential.
So the signal itself uses the stored energy. What's ATP's role then? If it's not powering the spark, what does it do?
It recharges the battery. After the impulses pass and those ions have moved across the membrane, slightly altering the concentrations, ATP is essential for the recovery phase.
The reset.
The reset, exactly. ATP-dependent active transport systems, the most famous being the sodium-potassium pump, although the source describes its function without necessarily naming it. Use the energy released from ATP hydrolysis to actively pump those ions back to where they started. Sodium gets pumped back out, potassium gets pumped back in, working against their gradients now.
Restoring the original situation.
Precisely. This restores the concentration gradients, reestablishing the stored potential energy across the membrane, effectively recharging the nerve fiber and making it ready to fire the next impulse. So ATP powers the pumps that maintain the ion gradients, the battery charge, not the brief discharge of the signal itself.
Wow, okay, so ATP is literally the workhorse fuel for building things, moving muscle, transporting molecules, getting stuff secreted, and resetting nerve signals. It's everywhere.
Yeah.
But as we touched on with muscle contraction, the demand can be huge and sudden. The ATP actually present in the cell, you said, might only last for a second or two of maximal effort.
That's right. The actual concentration of ATP floating around isn't enormous. It's enough for immediate needs, but not for sustained high demand.
So what's the immediate backup? The first line of defense when ATP starts getting used up fast.
This is where phosphocreatine comes in. It plays a critical role. While ATP is the primary direct currency, it's actually not the largest store of high-energy phosphate bonds readily available within the cell.
Oh, what is then?
Phosphocreatine is, the source calls it, an accessory storage depot.
Accessory storage depot, okay, like a nearby savings account.
A perfect description. It's a readily accessible reserve of high-energy phosphate, like having quick cash equivalents stashed very close by. And it's a significant reserve. In cells, especially muscle cells, which have high and variable energy demands, phosphocreatine is found in concentrations typically 3 to 8 times greater than the concentration of ATP itself.
3 to 8 times more abundant. Wow. And its high energy bond holds slightly more energy than ATP's. Didn't you mention that earlier?
Yes, that's a key feature. Under standard lab conditions, its bond energy is around 8,500 calories per mole. But again, under physiological conditions in the body, 37 degrees, typical reactant levels, its high-energy phosphate bond can release as much as 13,000 calories per mole when broken.
13,000 for phosphocreatine compared to ATP's 12,000 physiologically.
Exactly. That difference, that extra 1,000 to 1,500 calories per mole in potential energy, might seem small, but it's absolutely critical to how phosphocreatine functions as an energy buffer.
Okay, so how does it function? It doesn't directly power muscle contraction or anything itself, right?
Correct. That's crucial. Phosphocreatine cannot directly couple its energy release to power cellular functions like synthesis or transport in the way ATP can. Its job is much more specific.
What is its job then?
Its sole purpose is to rapidly transfer its high-energy phosphate group interchangeably with ATP. There's an enzyme called creatine kinase that facilitates this. The core reaction, which is reversible, is given in the source. Phosphocreatine plus ADP plus ATP plus creatine.
Okay, phosphocreatine plus ADP yields ATP plus creatine and vice versa. So when ATP is plentiful, like when the cell is resting, the reaction goes one way.
Yes. When ATP levels are high and ADP levels are consequently low, because ATP isn't being used much, the equilibrium of that reaction is pushed to the left. Energy from the abundant ATP is used to transfer a phosphate group to creatine, forming phosphocreatine. This effectively charges up the phosphocreatine store, building up this readily available reserve.
Like putting money into the savings account when you have extra cash.
Exactly.
And then when the cell becomes active and starts using ADP rapidly, converting it to ADP, the reaction reverses.
Precisely. When cellular activity increases, ATP gets hydrolyzed to ADP and phosphate. The concentration of ADP within the cell starts to rise. This increase in ADP concentration, combined with that slightly higher energy level inherent in phosphocreatine's bond compared to ATP's, powerfully drives the reaction to the right.
Pushing it towards making ATP.
Yes. The phosphate group and its associated energy are rapidly transferred from phosphocreatine to the newly formed ADP, quickly synthesizing new ATP molecules.
So that slight energy advantage of phosphocreatine acts like a constant pressure, always ready to refill the ATP supply the moment ADP levels start to creep up.
That's the essence of its role as the ATP buffer. It's a dynamic equilibrium. As long as there's a decent supply of phosphocreatine available, this rapid transfer ensures that the concentration of ATP within the cell is kept at an almost constant high level, even during those initial, sudden, intense bursts of energy demand, like the start of a sprint.
And keeping ATP levels stable is important.
Vitally important. As the source emphasizes, the rates of virtually all the key energy-dependent metabolic reactions in the cell are sensitive to the ATP concentration. If ATP levels were allowed to plummet every time you started exercising intensely, the entire cellular machinery would slow down drastically.
So phosphocreatine buys time.
It buys the cell precious time, maybe 5 to 10 seconds worth of maximal effort. It provides a rapid, immediate source of ATP replenishment, bridging the gap until the body's slower, but more sustainable energy production systems, like increased glycolysis and aerobic metabolism, can ramp up to meet the demand.
Okay, so we've got the immediate cache, ATP. We've got the rapid backup in the phosphocreatine savings account for maybe 5-10 seconds of intense demand. But neither of these are huge long-term stores, right? You can't run a marathon on just ATP and phosphocreatine.
Not even close. They're crucial for the start, but not for sustained effort.
So where does the energy come from for sustained activity lasting minutes or hours? And this is where oxygen really comes into play, isn't it?
Absolutely. This leads us directly to the two fundamental energy engines your body has, the anaerobic system and the aerobic system. The key distinction, as you hinted, boils down to one thing: oxygen or no oxygen.
Anaerobic energy derived without using oxygen.
Correct. Anaerobic pathways extract energy from food sources without requiring the simultaneous presence or utilization of oxygen. Aerobic pathways, conversely, can only derive energy from food with the utilization of oxygen through oxidative metabolism.
Okay. What food sources can power the anaerobic engine? Can fats or proteins be used anaerobically?
No, the source points out quite clearly that carbohydrates are the only significant macronutrient that can provide energy anaerobically to any appreciable extent.
Only carbs for anaerobic.
Oh,
Interesting.
yes. And the primary anaerobic process involving carbs is that first one we mentioned, glycolysis. The breakdown of glucose from the blood, or glycogen, stored glucose within the cells, into smaller molecules like pyruvic acid. In the absence of sufficient oxygen, this pyruvic acid is often converted further into lactic acid.
Right, lactic acid buildup during intense exercise. And what's the ATP yield like from this anaerobic glycolysis? Is it efficient?
It's fast, but not very efficient in terms of ATP yield per molecule of glucose compared to aerobic metabolism. For each mole of free glucose that enters the cell from the blood, that undergoes glycolysis all the way to pyruvic acid, you get a net yield of only 2 moles of ATP.
Just 2 ATP per glucose.
Okay. However, there's a slight bonus if you start with glycogen that's already stored within the muscle cell. Because glycogen is already in a partially activated or phosphorylated state, breaking down 1 glucose unit from glycogen via glycolysis yields a net of 3 moles of ATP.
Ah, 1 extra ATP if you start from stored glycogen. So that makes stored glycogen slightly better for anaerobic bursts.
Yes, exactly. The source explicitly states that stored glycogen within the cells is the best source of energy under strictly anaerobic conditions because it provides that slightly higher ATP yield per glucose unit, essentially skipping 1 initial energy investment step required for free glucose.
Okay, so anaerobic is carbs only, mainly via glycolysis, yielding 2 or 3 ATP per glucose. What about the aerobic fuels? Can everything be burned aerobically?
Pretty much. All 3 major macronutrients, carbohydrates, fats, and proteins, can be fully oxidized aerobically, meaning combined with oxygen, to generate large amounts of ATP. This happens through the pathways we mentioned earlier, like the citric acid cycle and the subsequent process called oxidative phosphorylation, which occurs primarily in the mitochondria and is where the vast majority of ATP is made when oxygen is available.
So the aerobic system is more versatile in fuel choice and produces much more ATP, but presumably it's slower. Why would the body ever choose to rely on the less efficient anaerobic system?
Speed and oxygen availability. The anaerobic system is crucial in situations where energy is needed faster than oxygen can be delivered to the muscles or utilized effectively by the aerobic metabolic machinery. The source describes 2 primary scenarios where anaerobic energy really shines, or rather becomes essential.
Okay, what's the first one?
First is acute hypoxia, a severe lack of oxygen. Imagine, heaven forbid, if someone stops breathing or is in an environment with very little oxygen. Your body has a very small, finite store of oxygen physically dissolved in body fluids, bound to hemoglobin in your blood, and bound to a similar molecule called myoglobin in your muscles. This stored oxygen can sustain aerobic metabolism for perhaps only about 2 minutes.
Only 2 minutes, wow.
Yeah, it's not much. Beyond that point, if external oxygen isn't available, your cells, particularly brain cells, which are very vulnerable, must rely on anaerobic glycolysis using their stored glycogen to generate the ATP needed to survive. This pathway can provide critical, life-sustaining energy for maybe another minute or so.
Generating lactic acid in the process.
Yes. Under these conditions, glycogen is broken down to pyruvic acid, which then accumulates as lactic acid because the subsequent aerobic pathway, the citric acid cycle, is shut down due to the lack of oxygen. It's a vital, though very short-term, survival mechanism when oxygen is critically limited.
Okay, so extreme oxygen deprivation. What's the second, maybe more common situation where anaerobic energy is key?
Strenuous bursts of activity. This is probably the most relatable example for most people. Think about maximal effort activities, sprinting as fast as you possibly can, lifting a maximum weight just once, performing a powerful jump.
All-out effort.
Exactly. Your aerobic oxidative metabolism is highly efficient at making ATP, but it takes some time to ramp up to its maximum rate, and it's ultimately limited by how quickly oxygen can be delivered by your heart and lungs and utilized by the mitochondria. For those first few seconds, up to maybe a minute or two, of truly maximal all-out physical effort, the demand for ATP is simply higher than the aerobic system can meet quickly enough.
The demand outstrips the aerobic supply rate.
Precisely. And the body has a layered strategy to cope with this, combining the energy systems we've discussed.
Okay, so walk us through that sequence. During, say, a 100-meter sprint, how does the body power that?
Right. The source lays out this sequence clearly. First, for maybe the first second or so, you use the tiny store of ATP already floating around in the muscle cells.
Just the immediate cache.
Yep. Then for the next, say, 5 to 10 seconds, you heavily relied on the phosphocreatine system. That stored phosphocreatine rapidly transfers its phosphate to ADP to regenerate ATP, keeping the ATP levels high during this initial burst.
Quick backup fund.
Exactly. Now, if the maximal effort continues beyond about 10 seconds and the intensity is still too high for the aerobic system to fully keep up, which it would be in a sprint, anaerobic glycolysis becomes the dominant energy provider.
Breaking down stored glycogen without oxygen.
Yes. This pathway, while less efficient overall, is significantly faster at producing ATP than the full aerobic oxidation pathway. It can provide the bulk of the energy for high-intensity activities lasting longer than 10 seconds, but generally less than about 1 to 2 minutes.
So phosphocreatine for the start. Then anaerobic glycolysis takes over for that middle phase of intense activity.
That's the general sequence for maximal bursts.
And relying heavily on that anaerobic glycolysis for, say, a minute of hard effort has consequences, doesn't it?
It absolutely does. The source highlights 2 main ones. First, you rapidly deplete the muscle's limited stores of glycogen. Second, the byproduct of anaerobic glycolysis, lactic acid, starts to accumulate significantly, both within the muscle cells and spilling over into the bloodstream. This contributes to muscle fatigue.
Which brings us neatly to what happens after you stop that sprint or intense effort. You're usually panting, breathing heavily for a while, right? That's the oxygen debt.
Exactly. You can't just stop breathing hard the moment you finish the intense anaerobic activity. Your body needs to consume a significant amount of extra oxygen above your normal resting oxygen consumption over the next several minutes, or even up to an hour or more for very strenuous exertion. This elevated post-exercise oxygen consumption is needed to recover and restore the systems that were pushed during the anaerobic phase.
And what is all that extra oxygen actually being used for?
Yeah.
Why do you need to keep breathing hard?
The source details several critical recovery tasks that require this extra oxygen. First, and perhaps the most significant part of the debt, a large portion of the accumulated lactic acid needs to be dealt with. A good chunk of it, maybe about 4/5, is transported via the blood to the liver.
Okay.
And in the liver, using oxygen-dependent aerobic metabolism, this lactic acid is converted back into glucose. This process is called gluconeogenesis.
So the lactic acid isn't just waste, it's a temporary holding pattern for carbon that can be recycled back into glucose fuel. That's efficient.
It is quite elegant, isn't it? The liver can then release this newly made glucose back into the blood for other tissues, like muscles, to use aerobically later, or it can be stored as glycogen in the liver itself. So oxygen is needed to power this recycling process.
Okay, dealing with lactic acid, What else is the oxygen debt used for?
Second, the extra oxygen supports the aerobic processes needed to reconvert all the ADP and AMP, adenosine monophosphate, which forms when two ADPs react. That built up during the intense activity back into ATP, refilling the cell's immediate energy currency reserves.
Restocking the ATP cache and the backup.
Yes. Third, oxygen is needed to aerobically generate the ATP required to convert creatine and phosphate back into phosphocreatine, rebuilding that crucial rapid access buffer store.
Replenishing the phosphocreatine savings account.
Got it.
Fourth, some oxygen is used simply to reestablish the normal pre-exercise concentrations of oxygen that were bound to hemoglobin in the blood and the myoglobin in the muscles. These act as small, readily available oxygen reserves that get depleted during intense work.
Resaturating the oxygen carrier.
Exactly. And fifth, a small amount is just needed to raise the oxygen concentration in the air within the lungs back to its normal resting level.
Just refilling the lungs, basically.
Right. All these processes, converting lactate, resynthesizing ATP and phosphocreatine, reloading hemoglobin and myoglobin, and restoring lung oxygen collectively require that sustained period of heavy breathing and elevated oxygen consumption after intense anaerobic activity stops. It's literally paying back the debt incurred by relying on the oxygen-independent systems during the burst. And the source mentioned this concept is important in athletics.
Very much so. The ability to tolerate the buildup of lactic acid and accumulate a large oxygen debt during performance, and then the efficiency with which an athlete can repay that debt during recovery periods or after the event is a key factor in performance and endurance in many sports involving repeated high-intensity bursts or sustained near-maximal effort.
Okay, so if we try to visualize the whole energy system now, like the diagram shown in the source figure 7301,
Yeah.
it really helps tie these pieces together.
It does. That figure provides a great overview. It clearly illustrates how the anaerobic pathways, primarily using glycogen and glucose, feed into ATP production, often generating lactic acid or pyruvic acid as byproducts when oxygen is limited.
Right.
Then it shows how the aerobic pathways, fueled by carbs, fats, proteins, and other intermediates like acetyl-CoA, are the major generators of ATP when oxygen is available, occurring mainly in the mitochondria.
And ATP is right in the middle.
ATP is central. The diagram shows its reversible link to the larger phosphocreatine store, the buffer system, and then it shows ATP ultimately powering all the different types of cellular work we discussed earlier. Synthesis, muscle contraction, active transport, nerve function, and so on.
And it reinforces that key functional difference. Anaerobic for the rapid, powerful bursts, aerobic for the sustained, efficient power.
Exactly. You can think of the anaerobic system's phosphocreatine first, then anaerobic glycolysis as being like booster rockets on a spacecraft. They provide massive power for a short duration, essential for getting off the ground or making quick, powerful maneuvers.
They're boosters.
While oxidative metabolism, the aerobic system, is like the main engine. It's slower to reach full throttle, but it's incredibly efficient and can generate huge amounts of ATP over long periods as long as fuel, especially fats, which are a large store, and oxygen are available. It just can't match the sheer rate of ATP delivery during those initial maximal bursts that the anaerobic boosters provide.
That's a great analogy. Okay, we've covered the energy molecules, ATP and phosphocreatine, and the different engines, anaerobic, anaerobic, for generating them quickly or for the long haul. Now, a critical question. How does the body actually control the speed of all this energy release? It clearly doesn't just run full throttle all the time, thankfully.
No, that would be incredibly wasteful and damaging. This is where the elegance of metabolic regulation comes in. Energy release, as we've seen, is orchestrated through complex series of chemical reactions, and virtually all biological reactions are catalyzed and controlled by specific proteins called enzymes.
Right, enzymes speed up reactions.
You do. The source reminds us of the basic principles of enzyme activity that govern the rate of these metabolic processes. An enzyme typically binds to a specific molecule, its substrate, changes it in some way, and releases a product, facilitating a reaction that might otherwise happen very slowly or not at all.
And the rate of that enzyme catalyzed reaction depends on things like how much enzyme there is and how much substrate there is.
Exactly. The basic relationship, sometimes described conceptually by what's known as Michaelis-Menten kinetics, is that the reaction rate is influenced by both the enzyme concentration and the substrate concentration. The source highlights a couple of different scenarios based on substrate levels using a helpful figure. figure 73:2.
Okay, what's the first scenario?
The first is when the substrate concentration is very high, much higher than the enzyme can process immediately.
Lots of raw material available.
Right. In that situation, there's so much substrate around that it effectively saturates all the available active sites on the enzyme molecules. Every enzyme molecule is busy working as fast as it possibly can. Under these conditions, the overall rate of the reaction is limited almost entirely by the concentration of the enzyme itself.
Adding more substrate won't help.
No, because the enzymes are already working at their maximum speed, Vmax. To increase the overall rate, you need to increase the amount of enzyme.
And the source gives that really clear example from the kidneys with glucose reabsorption in diabetes.
Yes, that's a perfect illustration. In uncontrolled diabetes, blood glucose levels can become extremely high. As the blood is filtered by the kidneys, this means the fluid entering the renal tubules also has a very high concentration of glucose, the substrate.
Right.
The transporter proteins, which act like enzymes in this context in the tubule walls, are responsible for reabsorbing this glucose back into the bloodstream. But there's a finite number of these transporters. When glucose levels are very high, these transporters become saturated. They're all working as fast as they can.
They hit their transport maximum.
Exactly. No matter how much more glucose is present in the filtrate, the rate at which it can be reabsorbed back into the blood is limited by the fixed number or concentration of these transporter enzymes proteins available in those cells. This is why excess glucose spills into the urine in uncontrolled diabetes. The transport system is overwhelmed, limited by enzyme concentration, and can't reclaim all the substrate.
Okay, so if substrate is high, enzyme amount is the bottleneck. What about the opposite situation when the substrate concentration is low?
When the substrate concentration is low enough that it's not saturating the enzyme, meaning there are plenty of free enzyme molecules available, the situation changes. In this case, the reaction rate becomes proportional to both the enzyme concentration and the substrate concentration.
So adding more substrate will speed things up.
Yes, generally. If you double the amount of substrate available, up to a point, the reaction rate will tend to double as well, assuming the enzyme concentration itself isn't limiting. The source gives examples like the absorption of some nutrients from the intestinal tract or the reabsorption of substances in the renal tubules when their concentrations in the fluid are naturally low. The rate of transport is directly related to how much of the substance is present because there are usually more than enough transporters, enzymes, to handle the small amount available.
Okay, that makes sense. Now, most metabolic energy pathways aren't just one step, right? They involve many reactions happening in sequence.
Absolutely. They are often long chains or cycles of enzyme-catalyzed reactions where the product of one step becomes the substrate for the very next step.
So how is the speed of the whole chain controlled?
In any linear series of reactions like this, the overall rate of the entire sequence, how quickly you get from the starting material to the final product, is generally limited by the rate of the slowest step in that series. Think of it like an assembly line.
The bottleneck.
Exactly. That slowest step is called the rate-limiting step. Speeding up other steps in the chain won't increase the overall output if the bottleneck step remains slow. To control the whole pathway's speed, the cell usually regulates the enzyme that catalyzes this rate-limiting step.
Okay, so in the complex chains of reactions that break down food to make ATP, there must be a key rate-limiting factor, a master control switch that governs the overall speed of energy release for the whole cell. What is that master control?
The source is quite definitive on this. It identifies the major rate-limiting factor for almost all energy metabolism in the body, both anaerobic and aerobic pathways leading to ATP synthesis as the concentration of ADP, adenosine diphosphate.
ADP, the molecule that's left over after ATP is spent. How does that control things?
It's a beautifully simple and effective feedback mechanism. Think about it. When a cell is at rest, its energy demand is low. ATP is not being rapidly broken down to ADP and phosphate. Therefore, the concentration of ADP within the cell is kept very, very low.
Okay, low ADP at rest.
Now, ADP happens to be a required substrate or an essential activator for several key enzymes involved in the major ATP-generating pathways, particularly enzymes in glycolysis and, very importantly, the process of oxidative phosphorylation in the mitochondria, where most ATP is made aerobically.
So if ADP is scarce?
These pathways run run very slowly. There's simply not enough of one of their key inputs, ADP, to keep them going quickly. The cell isn't demanding much energy, so it doesn't need to produce much ADP, and the low ADP level automatically keeps production low.
But then when the cell becomes active, say, a muscle cell starts contracting, it starts using ATP rapidly.
Right. Activity means ATP gets broken down into ADP and phosphate. This causes the concentration of ADP inside the cell to increase.
More ADP appears.
And this increase in ADP concentration acts as a powerful activating signal. Higher ADP levels automatically stimulate and increase the rates of those key rate-limiting enzymes in all the major energy-releasing metabolic pathways. Glycolysis speeds up, the citric acid cycle speeds up, beta-oxidation of fat speeds up, and oxidative phosphorylation ramps up everything that ultimately feeds into making more ATP.
Wow. So the cell's own activity level, reflected in how much ADP it produces, directly controls how fast its energy engines run.
It's an incredibly elegant and efficient feedback loop. More cellular work leads to more ATP breakdown, which means more ADP is present. This rise in ADP then directly signals the energy-producing machinery to speed up and replenish the ATP supply. Conversely, when the cell's activity decreases again, ATP is used less rapidly. ADP levels quickly drop as the existing ADP gets converted back into ATP by the now slowing pathways.
And the low ADP automatically slows down energy production again.
Exactly. The energy-generating pathways automatically slow down or even stop because their required substrate or activator, ADP, is no longer abundant. The rate of energy release is precisely and automatically controlled by the cell's own minute-to-minute demand, as directly signaled by the concentration of ADP. It's a perfect example of matching energy supply to energy demand in real time, without needing complex external signals for this basic regulation.
That system of control at the cellular level, driven by ADP, is just brilliant in its simplicity and effectiveness. Okay, now that we see how the rate is regulated within individual cells, let's zoom out again to the whole body and talk about the overall rate of energy expenditure for the entire person, the metabolic rate.
Right, scaling up from the cell to the organism. Metabolism, broadly speaking, refers to the sum total of all the chemical reactions occurring in all the cells of your body at any given time.
All the chemistry of life.
Pretty much. And the metabolic rate is simply the rate at which the sum total of reactions is happening. The standard way to measure and express this overall metabolic rate is by quantifying the rate of heat liberation from the body.
Okay, why heat? We touched on this briefly, but why is heat the standard measure of metabolic rate?
Because, as the source makes very clear, heat is the ultimate end product of almost all the energy that is released and transformed within the body through these metabolic processes. When your body breaks down food, makes ATP, and then uses that ATP, energy is constantly being converted from one form to another.
And conversions aren't perfectly efficient.
Never perfectly efficient. Some of that energy might be transiently stored in chemical bonds, like ATP itself, or in concentration gradients across membranes, but a significant portion is always released as heat at various stages of these conversions due to the laws of thermodynamics.
But even during the process of making ATP itself.
Yes. The source estimates that, on average, about 35% of the chemical energy originally present in the food you eat is immediately released as heat during the process of forming ATP. So right off the bat, more than a third is lost as heat just in capturing energy in ATP bonds.
Okay, 35% lost initially. What about the energy that is captured in ATP and then used for work? Does that also become heat?
Overwhelmingly, yes. The source states that even under the most optimal conditions, probably no more than about 27% of the total energy originally derived from food is finally utilized by the functional systems of the cells for doing things like muscle contraction, active transport, synthesis, etc.
Only 27% used for function. So the other 73% or more eventually becomes heat.
That's the bottom line. And even that 27% portion that is used for cellular functions still ultimately ends up being converted into heat within the body, unless that energy is used to perform external work.
How does the energy used for, say, building proteins end up as heat?
Good question. Consider protein synthesis. ATP energy is used to form peptide bonds, effectively storing some energy in the protein structure. But your body's proteins aren't static. They are constantly being broken down and rebuilt in a process called protein turnover. When those proteins are eventually degraded, the energy that was stored in their peptide bonds is released, and guess what form it takes?
Heat.
Heat. Similarly, think about muscle activity. A lot of the energy used by contracting muscles goes into overcoming the internal friction and viscosity of the muscle tissues and surrounding structures as they move. This friction generates heat directly within the body.
Like rubbing your hands together.
Exactly. Or consider the heart pumping blood. The heart uses ATP energy to create pressure, pushing blood into the arteries. Some of that energy is temporarily stored as potential energy in the stretched elastic walls of the arteries. But as the blood flows through the miles of blood vessels, friction between the blood and the vessel walls dissipates this potential energy entirely as heat.
So unless you're actually lifting something or climbing stairs, all the energy ends up as heat inside you.
Precisely. The only significant exception is when your muscles perform external work against gravity or some other external force, like lifting a heavy object, climbing a hill, or cycling against resistance. In those cases, some of the energy is transferred to the external environment as work. But even then, a large fraction is still dissipated as heat within your body due to inefficiencies in muscle contraction itself.
So measuring the total heat produced by the body is a very accurate way to determine its total metabolic activity.
It is indeed, because essentially all the energy released by your metabolism is eventually converted into heat within your body, unless external work is done. Measuring the rate of heat liberation provides a direct and comprehensive measure of your overall metabolic rate.
Okay, now to talk numbers here, we need to be absolutely clear on the unit of energy. The source really emphasizes using the calorie with a capital C. Can you clarify that difference again?
Yes, this is a frequent point of confusion, but absolutely critical for understanding nutrition and metabolism. A calorie, spelled with a small c, is a gram calorie. It's defined as the amount of heat required to raise the temperature of 1 gram of water by 1 degree Celsius. This is a very, very small unit of energy.
Too small for body energy.
Way too small for practical use in physiology or nutrition. The unit we see on food labels, and the one universally used when discussing body energy metabolism, is the calorie with a capital C. This is actually a kilocalorie, kcal.
Kilo, meaning 1,000.
Right. 1 calorie, capital C, is equal to 1,000 calories, small c. It's the amount of heat needed to raise the temperature of 1 kilogram at 1,000 grams of water by 1 degree Celsius. This larger unit is much more appropriate for the scale of energy transfer happening in the human body. So whenever we talk about metabolic rate or food energy, always remember it's calories with a capital C, meaning kilocalories.
Got it. Capital C calories it is. So the overall metabolic rate is measured by the rate of heat production in these calories, kilocalories. How do scientists actually measure this for a living person? It sounds tricky.
It can be. The source describes 2 main methods. The most direct way, conceptually at least, is called direct calorimetry.
Direct calorimetry, as the name implies, measuring the heat directly.
Exactly. You literally measure the heat coming off the person. The subject is placed inside a specially constructed chamber, basically a very well-insulated room, that prevents any heat from escaping to the outside or entering from the outside.
Like a human-sized thermos.
Ah, something like that. The heat produced by the person's metabolism naturally warms the air inside this chamber. This warmed air is then circulated, often through a system of pipes that are immersed in a precisely measured bath of water. By carefully measuring the rate at which the temperature of the water bath increases, scientists can calculate the rate at which heat was transferred from the person's body to the water. This rate of heat transfer directly equals the person's metabolic rate, their rate of heat liberation.
That sounds like quite an elaborate setup. Is it used often?
It is elaborate. Direct calorimetry chambers are large, complex, and very expensive to build and operate. The source notes that performing measurements this way is physically difficult and time-consuming, so it's typically used only in specialized research settings, not for routine clinical assessments or large-scale studies.
Okay, so direct measurement is possible but impractical for most uses. What's the more common practical method then?
The much more widely used method is indirect calorimetry. As the name suggests, this method calculates the metabolic rate indirectly, not by measuring heat itself, but by measuring the person's rate of oxygen consumption.
And this works because, as we discussed, most of the body's energy production uses oxygen, right?
Precisely. The fundamental principle is that, as the source points out, over 95% of the energy expended by the body under normal conditions is derived from metabolic reactions that involve the consumption of oxygen to oxidize food substances, carbs, fats, proteins.
So oxygen use is a good proxy for energy release.
It's a very good proxy. By carefully measuring the volume of oxygen a person consumes over a specific period, using devices that measure inhaled and exhaled air composition and volume, you can accurately estimate the rate of their oxidative metabolism. And from that, you can calculate the amount of heat energy liberated.
Is there a simple conversion factor for that, like X calories released per liter of oxygen consumed?
Yes, there is. The key concept here is the energy equivalent of oxygen. This represents the average amount of energy liberated as heat per liter of oxygen consumed. Now, this value varies slightly depending on exactly what fuel is being burned.
Because burning carbs versus fats might yield different energy per oxygen molecule used.
Exactly. The source gives the specific values. When your body metabolizes pure glucose, using 1 liter of oxygen yields about 5.01 calories of energy. For starches, it's very similar, about 5.06 col. For fat, it's a bit lower, around 4.70 col. And for protein, it's the lowest of the three, about 4.60 col.
So they're all clustered around, what, 4.6 to 5.1 calories per liter of oxygen?
Right. And for someone eating a typical mixed diet containing carbs, fats, and proteins, the average energy equivalent of oxygen is generally taken to be about 4.825 calories per liter.
Okay, 4.825 col on average.
So in practice, indirect calorimetry involves measuring the volume of oxygen consumed by the person over a set time period, say liters per minute or liters per hour, and then multiplying that oxygen consumption rate by this average energy equivalent, 4.825 col, to get a good estimate of their total energy expenditure rate, metabolic rate, in calories per minute or calories per hour.
How accurate is using that single average value if someone's diet might be high carb or high fat at that moment?
It's usually quite accurate for most purposes. The source notes that if a person happened to be metabolizing only carbohydrates during the measurement, using the average 4.825 value would slightly underestimate their actual energy expenditure by about 4%, because the true value for carbs is a bit higher, 5.01.
Okay.
Conversely, if they were burning exclusively fat during the test, using the average would slightly overestimate their energy expenditure by about 4%, since the true value for fat is a bit lower, 4.70. But for people eating a normal mixed diet, the variations tend to cancel out, and the calculation using the average energy equivalent is typically accurate to within just a few percent of the true metabolic rate. More sophisticated calculations can account for protein metabolism by measuring nitrogen excretion, but for many applications, just measuring oxygen consumption is sufficient.
Okay, so we can measure overall metabolic rate pretty well using oxygen consumption via indirect calorimetry. Now let's get into the factors that actually determine how high or low a person's metabolic rate is on any given day. What accounts for the big differences we see in how many calories people seem to need?
Right, this is where individual variability really comes into play. The source first establishes the concept of energy balance. For a healthy adult who is maintaining a stable body weight, their total energy intake from food over a period must, on average, equal their total energy expenditure.
Intake equals output for weight stability. Makes sense.
And just for context, the source mentions that the average American diet at the time of writing was estimated to be roughly 45% carbohydrate, 40% fat, and 15% protein. These are the inputs.
Okay, what about the outputs? What are the main components of daily energy expenditure?
The source breaks down the total daily energy output into several key components. The four main areas generally considered are, first, the basal metabolic rate, or BMR.
BMR, we'll come back to that. What else?
Second, the energy used for all kinds of physical activities. This includes planned exercise, but also all the spontaneous movements of daily life.
Activity energy. Got it.
Third, the thermic effect of food, or TEF, which is the energy cost of processing the food you eat.
TF. Okay.
And fourth, sometimes considered separately but often intertwined with the others, is the energy required specifically for maintaining body temperature, particularly in response to cold, through processes called thermogenesis.
Okay. BMR, activity, TEF, and temperature regulation. Let's start with the biggest baseline component, BMR. What exactly is the basal metabolic rate?
The basal metabolic rate is defined as the absolute minimum rate of energy expenditure required by the body to maintain its essential physiological functions when you are in a state of complete physical and mental rest, in a thermally neutral environment, and in a post-absorptive state, meaning you haven't eaten recently.
So the energy cost of just existing, keeping the lights on.
That's a perfect way to put it. It's the energy needed for your heart to keep beating, your lungs to keep breathing, your brain and nervous system to maintain basic function, your kidneys to filter blood, and for all your cells to carry out essential maintenance activities like maintaining ion gradients across their membranes. Even at complete rest, your body is humming along using energy.
And how much of our daily energy does BMR typically account for?
It's actually the largest single component for most people, especially those who are relatively sedentary. The source estimates that BMR accounts for roughly 50% to 70% of the total daily energy expenditure in most individuals.
Half to nearly 3/4.
Yeah.
Just for baseline function. Wow.
It's substantial. And measuring BMR is particularly useful for comparing metabolic rates between different individuals or tracking changes in one person over time because the measurement conditions are so strictly controlled.
Right. You mentioned strict conditions. Right. What are they again? Why are they so important?
They're crucial because they aim to eliminate or minimize all the other factors that can influence metabolic rate, leaving just the true baseline. The source lists them precisely. First, the person must not have eaten for at least 12 hours. This ensures they are post-absorptive, eliminating the variable energy cost of digesting and processing food, the TE.
Okay. 12-hour fast.
Second, the measurement should be taken after a full night of restful sleep. This helps ensure the muscles are relaxed and the nervous system isn't overly active from lack of sleep. Third, no strenuous exercise or significant physical activity should have been performed for at least an hour before the test, preferably longer. Activity raises metabolic rate for some time afterwards.
No recent exercise.
Fourth, the environment must be thermally neutral, a comfortable room temperature, typically cited as between 68 degrees and 80 degrees, or 26.7 degrees C. This prevents the body from having to expend extra energy on either generating heat to stay warm or actively cooling itself down.
Comfortable temperature.
And fifth, and critically, the person must be completely at rest, both physically, usually lying down, and mentally calm, not anxious or excited during the actual measurement period. Any muscle tension or mental agitation will increase energy use.
Total rest during the test.
Meeting all these conditions simultaneously allows for the measurement of the lowest rate of energy expenditure compatible with wakeful life, the true basal metabolic rate.
What's an average BMR value for, say, an adult man?
For a typical 70 kilogram, about 154 pound adult man, the BMR is usually around 65 to 70 calories per hour. That works out to about 1560 to 1680 calories over a full 24 hours, just for baseline function.
So over 1,600 calories a day just lying still.
For an average man, yes. And what specifically is contributing to that minimum rate? A large part, as we said, is the energy needed to keep the vital organs running, the central nervous system, brain and spinal cord, the heart constantly pumping, the kidneys filtering, the liver performing its myriad metabolic tasks.
Okay. But when you compare BMR between different individuals, what causes the variation? Why might one person's BMR be higher or lower than another's, even if they are the same age and sex?
The source points to two primary factors driving individual differences in BMR, assuming other factors like hormones are normal. The biggest ones are the amount of lean body mass, particularly skeletal muscle mass, and overall body size.
Skeletal muscle. Even when it's at rest, it uses a lot of energy.
It absolutely does. Resting skeletal muscle, even when you're completely still, is metabolically quite active. It's constantly maintaining tone, turning over proteins, pumping ions. The source estimates that resting skeletal muscle accounts for about 20% to 30% of the entire BMR. So someone with more muscle mass will generally have a higher BMR than someone with less muscle mass, even at the same total body weight.
More muscle burns more calories, even at rest. That's important.
Very important. And overall body size matters too, partly because larger bodies have more tissue mass overall, but also because BMR is related to body surface area. Larger surface area tends to mean greater heat loss to the environment, which needs to be compensated for metabolically. For better comparison across individuals of different sizes, BMR is often normalized by body surface area and reported in units like calories per hour per square meter, CalNorm.
Okay. Looking at that figure in the source, figure 73-4, that shows BMR trends, we also see clear effects of age and sex, right?
Yes, that figure illustrates two major population trends. First, BMR tends to decline progressively as people get older, particularly in adulthood.
Why does it decline with age?
This decline is likely associated with several factors, but a major one is the natural tendency for skeletal muscle mass to decrease and body fat percentage to increase as people age, a condition called sarcopenia. Since muscle tissue is much more metabolically active than fat tissue, this shift in body composition leads to a lower overall BMR. Changes in hormonal activity might also play a role.
Okay, age lowers BMR, partly due to muscle loss. What about sex?
The figure also shows that, on average, women generally have slightly lower BMRs than men of the same age and size. Again, this difference is often attributed primarily to differences in average body composition. Women typically have a lower percentage of skeletal muscle mass and a higher percentage of body fat compared to men, which results in a lower average basal metabolic rate. Hormonal differences might also contribute to some extent.
So baseline BMR depends on size, muscle mass, age, and sex. But beyond these fundamental factors and general trends, the source lists several other specific things that can act like metabolic switches or regulators, significantly altering BMR or overall metabolic rate.
Yes, this is where factors like hormones and physiological state come in, and some of them can have really dramatic effects. The single most powerful hormonal factor mentioned as influencing metabolic rate is thyroid hormone, specifically thyroxine, T4, and its active form triiodothyronine, T3.
Thyroid hormone. How much impact can it actually have?
Big, profound impact. The source states that when the thyroid gland is maximally stimulated to secrete large amounts of thyroxine, the overall metabolic rate can increase by as much as 50% to 100% above the normal level.
Double the normal rate.
Wow. Yes. It essentially acts like a general accelerator pedal for chemical reactions in almost all cells throughout the body. Conversely, if thyroid function is completely lost, for example, due to disease or removal of the gland without hormone replacement, the metabolic rate can plummet dramatically, falling to 40% to 60% below the normal level. This highlights just how critical thyroid hormone is for maintaining normal metabolic pace.
And the source also mentioned a link between thyroid function and climate adaptation.
It did. It noted that people living in very cold climates tend to have BMRs that are 10% to 20% higher than those living in tropical regions. This is thought to be partly due to chronic cold exposure and stimulating increased secretion of thyroid hormone, which helps generate more body heat.
Interesting adaptation. Are there any other hormones with significant effects on metabolic rate?
Yes, a few others are mentioned. Male sex hormone, testosterone, can increase the metabolic rate, typically by about 10% to 15% above what it would otherwise be. This effect is thought to be largely indirect, resulting primarily from testosterone's known effect of increasing skeletal muscle mass, which, as we discussed, has a higher metabolic rate.
So more muscle from testosterone leads to higher BMR.
That seems to be the main mechanism. Female sex hormones, like estrogen, might cause a slight increase in metabolic rate at certain times, but the effect is generally considered much less significant or consistent compared to thyroid hormone or testosterone.
Okay, what about growth hormone?
Growth hormone also increases metabolic rate. It does this both by directly stimulating cellular metabolism in various tissues and, similar to testosterone, by promoting the synthesis of proteins, particularly increasing muscle mass. The source provides a clinical example. In adults who have growth hormone deficiency, initiating growth hormone replacement therapy can increase their BMR by around 15% to 20%.
So thyroid, testosterone, and growth hormone can all boost metabolism.
Yeah.
What about something completely different, like having a fever? Does being sick and hot speed things up?
Absolutely. Fever has a very direct and predictable effect on metabolic rate. Regardless of what's causing the fever, infection, inflammation, etc., the simple fact of having an elevated body temperature directly increases the speed of almost all chemical reactions occurring throughout the body. Enzymes just work faster at higher temperatures up to a point.
How much faster?
The source gives a rather dramatic quantification based on the Van Hofer-Arrhenius relationship for chemical reaction rates. It states that, on average, the rate of cellular chemical reactions increases by about 120% for every 10 degrees C rise in body temperature. Or, put another way, roughly a 13% increase in metabolic rate for every 1 degree, about 7% per 1 degree C, rise in body temperature above normal.
13% per degree Celsius. That's significant.
It is. So if you have a fever of, say, 39 degrees C, about 102.2 degrees alpha, which is 2 degrees C above normal 37 degrees C, your metabolic rate could be roughly 26% higher than usual just due to the fever itself. We'll touch more on temperature regulation later, but the impact of fever on boosting metabolism is undeniable.
Makes sense. Now what about sleep? We measure BMR after restful sleep, but does sleep itself lower metabolism compared to just resting awake?
Yes, it does. During periods of sleep, the overall metabolic rate typically decreases, falling about 10% to 15% below the normal waking basal metabolic rate.
Why does it drop during sleep?
The source suggests two main reasons. First, there's a significant decrease in skeletal muscle tone during most stages of sleep. Your muscles become much more relaxed than even when you're resting awake. Second, there's a general decrease in the activity of the central nervous system during sleep compared to the alert waking state. Both these factors contribute to the lower energy expenditure during sleep.
Okay. And finally, the source mentioned malnutrition as a factor that lowers BMR.
Yes, prolonged or severe malnutrition can cause a significant decrease in metabolic rate, sometimes dropping at 20% to 30% below normal levels.
Why does starvation slow metabolism down?
The source explains this is likely due to a general paucity of food substances available within the cells. Essentially, when the body is chronically deprived of energy and nutrients, there's a lack of the necessary substrates, vitamins, and cofactors required for metabolic reactions to proceed at their normal rate. The body seems to enter a state of conservation, slowing down non-essential processes to save energy.
A survival mechanism, perhaps.
Likely. The source also notes the grim consequence that in the final stages of many wasting diseases associated with severe malnutrition, this marked decrease in metabolic rate can become so profound that the person's body temperature may actually fall significantly below normal just before death.
That's sobering. Okay, so BMR sets the essential baseline, and it's influenced by our fundamental makeup, size, muscle, age, sex, and significantly modulated by hormones: thyroid, sex hormones, growth hormone, body temperature, fever, sleep state, and nutritional status. But the biggest day-to-day variation in total energy expenditure usually comes from physical activity, right?
Absolutely, hands down. While BMR represents the fairly stable energy needed for ascended functions at rest, any form of physical activity layered on top of that dramatically increases the demand for energy. And this component is highly variable between individuals and from day to day for the same person.
And the source gave some truly astonishing numbers earlier for how much muscle activity can potentially increase energy use, didn't it?
It did. It mentioned that a single muscle fiber performing a short maximal contraction can increase its rate of energy expenditure by up to 100 times compared to its resting rate, even if only for a few fractions of a second.
100-fold increase at the fiber level.
And for the whole body, engaging in maximal muscle exercise, think an Olympic sprinter or weightlifter during peak effort can increase the overall rate of heat production, metabolic rate, to about 50 times the normal basal level, although only sustainable for very short periods. For more sustained, heavy exercise like marathon running or intense labor, a well-trained individual might sustain a metabolic rate around 20 times their normal basal rate for hours.
50 times normal for bursts, 20 times for sustained heavy work. That huge potential range of increase is precisely why physical activity is the single most important factor causing the wide variations we see in people's total daily calorie needs.
Exactly. A very sedentary person might get by on, say, 2,000 calories per day, with their BMR making up the largest chunk of that. But someone engaged in consistently heavy physical labor or intense athletic training might easily require 6,000, 7,000, or even more calories per day. That threefold or greater difference is almost entirely due to the energy expended during their physical activities.
And the source explicitly connected this to the challenge of weight management in modern life.
Yes, it makes the point that in many industrialized countries, abundant and palatable food makes high energy intake relatively easy, while lifestyles have often become much more sedentary, leading to low energy expenditure from physical activity. This common mismatch, high intake, low output, results in a positive energy balance, where the excess energy is stored primarily as fat, contributing to the high rates of overweight and obesity. It strongly underscores the critical importance of incorporating regular physical activity for maintaining energy balance and health.
And physical activity isn't just structured exercise like going to the gym or for a run, is it?
No, definitely not. The source implicitly includes what exercise physiologists often call non-exercise activity thermogenesis, or NEAT.
NEAT? What does that cover?
NEAT encompasses all the energy expended for physical activities that are not deliberate exercise sessions. This includes things like the energy cost of maintaining posture while sitting or standing, spontaneous movements like fidgeting, walking around the office or house, doing chores, taking the stairs instead of the elevator, even shivering when cold.
All the little movements of daily life.
Exactly. For a very sedentary person, NEAT might account for only a relatively small percentage of their daily energy expenditure, maybe around 7% according to the source's general figures. However, NEAT is highly variable between individuals. Two people with similar office jobs might have vastly different NEAT levels depending on their habits. One might constantly fidget and walk around, while the other sits very still. These differences in NEAT can actually contribute significantly to variations in overall daily energy expenditure and susceptibility to weight gain. Your occupation also plays a huge role here. Obviously, a construction worker's NEAT is vastly higher than a desk worker's.
That's a really important point. All movement counts. Looking back at that table 73301, and the source, the one showing energy expenditure for different activities, really brings home how much the intensity matters.
Oh, absolutely. Those specific numbers, even if they are averages for a 70-kilogram person, are powerful because they make the differences so concrete and memorable.
Can you give a few examples from the table?
Sure. Sleeping is listed at about 65 calories per hour. Just lying still but awake uses a bit more, 77 calories. Sitting quietly is around 100 calories. Walking slowly, about 2.5 mile per hour, uses 200 calories. Now compare that to running at a moderate pace, like a 10-minute mile, which burns about 570 calories.
Okay, running is much higher.
But then look at something really intense, like walking upstairs rapidly. The table lists that at a whopping 1,100 calories per hour.
1,100. That's almost 17 times more than sleeping.
Exactly. These values vividly illustrate the dramatic difference that the type and intensity of physical activity make to your instantaneous energy expenditure rate. It really emphasizes why activity is the most controllable and impactful part of the energy output equation for most people.
Okay, so BMR is the foundation. Physical activity, including NEAT, is the big variable layer on top that determines most of the difference in daily needs. What about the third component? You mentioned the energy used just to process the food we eat, the thermic effect of food.
Right, the thermic effect of food, or TFEF. Sometimes it's also called diet-induced thermogenesis, or the specific dynamic action, especially for protein. This refers to the fact that your metabolic rate actually increases for a period of time after you consume a meal.
Why does eating cost energy?
It's the energy required for the processes involved in handling the food. Digestion, secreting enzymes, contracting gut muscles, absorption of the nutrients across the gut wall, transporting those nutrients to the tissues, and then metabolically processing and storing those nutrients within the cells. All these steps require energy, primarily from ATP, and this metabolic activity generates extra heat.
Like the body has to work to handle the incoming fuel.
Exactly. It's an obligatory energy cost associated with processing your meals.
How much does TEEF typically add to the total daily energy expenditure?
The source suggests that, on average, TFIF accounts for roughly 8% of a person's total daily energy expenditure. So if someone eats 2,500 calories a day, about 200 of those calories might be used up just in processing that food.
About 8%. Does it vary depending on what kind of food you eat?
Yes, it does vary somewhat depending on the macronutrient composition of the meal. Meals that are high primarily in carbohydrates or fats generally cause a relatively small increase in metabolic rate due to TEEF. Perhaps around 4% or so of the energy contained in those carbs or fats is expended in processing them.
Okay, carbs and fats have a fairly low TEEF, but protein is different, isn't it?
Yes, protein has a uniquely high thermic effect. This is often referred to specifically as the specific dynamic action SDA of protein. After consuming a meal rich in protein, the metabolic rate typically starts to rise within about an hour, can reach a peak increase of maybe 20% to 30% above the basal metabolic level, and this elevated rate can persist for considerable time, sometimes 3 to 12 hours.
30% increase just from protein. Why is protein so much more costly to process?
It's thought to be related to the complex steps involved in metabolizing amino acids deamination, removing the nitrogen group, converting the carbon skeletons into usable intermediates, synthesizing urea to dispose of the nitrogen. These processes are apparently more energetically expensive than the initial steps of carbohydrate or fat metabolism.
Interesting. So that higher T for protein contributes to why it's often considered more satiating, perhaps.
That's certainly part of the thinking, yes. The higher energy cost and prolonged metabolic effect might contribute to feelings of fullness and potentially slightly increase overall energy expenditure compared to isocaloric meals lower in protein.
Okay, so we have BMR, activity, and TF. The last factor you mentioned was related to maintaining body temperature. Is there any other way the body intentionally generates heat besides shivering, which is muscle activity, or just as a byproduct of T?
Yes, there is a mechanism called non-shivering thermogenesis. As the name implies, this is a way the body can generate extra heat, specifically in response to cold stress, without involving the overt, rhythmic muscle contractions of shivering.
Non-shivering heat production. How does that work?
It's primarily stimulated by activation of the sympathetic nervous system part of your fight or flight response, which also gets activated by cold. The sympathetic nerves release hormones like norepinephrine, noradrenaline, and epinephrine, adrenaline, into the bloodstream and directly onto certain target tissues.
Okay.
These hormones then act on cells to directly increase their metabolic activity and rate of heat production independent of muscle contraction.
And is there a special tissue involved in this? You hear about brown fat sometimes?
Yes. Brown adipose tissue, or BAD, is a type of fat tissue that is particularly specialized for non-shivering thermogenesis. It looks different from the typical white adipose tissue, WAT, that stores most of our body fat.
How is brown fat different?
Unlike white fat cells, which mainly contain one single large droplet of stored fat, brown fat cells contain numerous mitochondria, those energy-producing organelles, and many small lipid fat droplets scattered throughout the cytoplasm. The high concentration of mitochondria, which contain iron, gives the tissue its characteristic brownish color.
Lots of mitochondria. And those mitochondria in brown fat are key to the heat production.
Absolutely crucial. The source explains that the mitochondria in active brown fat have a unique property. There's a special protein called uncoupling protein 1, UCP1, in their inner membrane. When brown fat is stimulated by the sympathetic nervous system via norepinephrine, UCP1 essentially allows the energy released during the oxidation of fuel, like fatty acids, to dissipate directly as heat instead of being efficiently captured to produce ATP.
So it uncouples fuel burning from ATP production.
Exactly. The normal coupling between the electron transport chain, where fuel is oxidized, and ATP synthesis, oxidative phosphorylation, is deliberately bypassed or relaxed in stimulated brown fat. As a result, almost all the energy released from burning fuel in those mitochondria gets released immediately as heat. Brown fat acts like a dedicated metabolic furnace designed specifically for rapid heat generation when needed.
That's fascinating. How important is this brown fat furnace in humans? Do adults have much?
The source notes that brown fat is much more abundant and physiologically significant in newborns, neonates. It plays a vital role in helping babies, especially premature ones, maintain their body temperature after birth, as they have a high surface area to volume ratio and can lose heat easily. In infants, maximal sympathetic stimulation of brown fat can more than double their overall metabolic rate, generating substantial heat.
Okay, important for babies. What about adults?
In adult humans, the amount of active brown fat is typically much lower, often found in specific locations like around the neck and upper back. Its contribution to overall non-shivering thermogenesis in response to cold is generally estimated to be relatively small, perhaps less than 15% of the total metabolic increase, although there's evidence it might become slightly more active or increase somewhat with prolonged or repeated cold exposure, cold adaptation. So while present and functional, it's usually not considered a major player in adult thermoregulation compared to shivering or behavioral changes.
Okay. But the source mentioned a potential link between this type of thermogenesis, maybe involving brown fat or sympathetic activity, and obesity.
Yes, this is an area of ongoing research that the source touches upon. There's some evidence suggesting that increased non-shivering thermogenesis, possibly mediated by increased sympathetic nervous system activity affecting brown fat, or perhaps even white fat browning, might serve as a partial buffer against weight gain in some individuals.
How would that work?
The idea is that in response to chronic excess caloric intake, the sympathetic nervous system might become more active, potentially influenced by hormones like leptin signaling energy excess to the brain, specifically the hypothalamus. This increased sympathetic drive could then stimulate thermogenesis, essentially causing the body to waste or burn off some of the excess energy intake as heat, thereby limiting the amount that gets stored as fat.
Like a metabolic safety valve.
Potentially, yes. It could represent one of the body's often insufficient defense mechanisms against developing obesity in an environment where high-calorie food is readily available and overconsumption is easy. However, the extent to which this mechanism operates effectively in humans and how much it varies between individuals is still being actively investigated. It's likely only one small factor among many influencing weight regulation.
Still a very interesting possibility. Wow. We have really covered a lot of ground today, peeling back the layers on energy from the fundamental currency inside the cell all the way out to how we measure and understand the whole body's energy expenditure and what controls it.
We really have. We started with ATP as the immediate cellular cache, saw how phosphocreatine acts as that crucial rapid backup buffer. Then we explored the two main engines, The fast but limited anaerobic system fueled primarily by carbs for those quick bursts, and the highly efficient, versatile aerobic system powered by carbs, fats, and proteins for sustained activity as long as oxygen is available.
And we saw the sheer elegance of a body's primary control system for energy release, where the concentration of ADP, the byproduct of energy use, acts as the master switch, automatically and perfectly linking the speed of energy production directly to the cell's real-time energy needs.
It's beautiful feedback control. Then we zoomed out to the whole body metabolic rate, understanding why heat is the ultimate measure, clarifying the crucial difference between calorie and calorie, always use the capital C, and looking at how it's measured either directly with calorimetry or, more commonly, indirectly using oxygen consumption and the energy equivalent of oxygen.
And finally, we delved into all the factors that influence that overall metabolic rate, starting with the basal metabolic rate, the baseline energy cost of living, which is shaped by our fundamental body size, muscle mass, age, and sex.
Right, and then layered on top of that are the powerful effects of hormones like thyroid hormone, the major accelerator, testosterone, and growth hormone. Plus the effects of body temperature itself, like during a fever, the dip during sleep, and the impact of nutritional status like malnutrition.
And then, of course, the most variable and often largest factor, the energy costs of physical activity, including both deliberate exercise and all the non-exercise activity of daily life, along with the energy cost of processing our food, TEF, especially protein, and the specialized non-shivering thermogenesis involving brown fat. It's a complex interplay.
It truly is.
And this deep dive into your internal energy system, understanding all these components, isn't just academic theory. It helps you understand, really at a foundational, professional level, why your body needs the fuel it gets from food, how it manages those inputs second by second, why different activities burn energy at vastly different rates, why things like sleep, hormones, or even just fidgeting affect your overall energy balance, and how this incredibly complex system constantly adapts to what you're doing and the environment you're in. It gives you a profound insight into the engine that powers your very existence.
Yeah, it's easy to just live our lives and take for granted the feeling of being alive and having the energy to move, think, and interact with the world. We don't consciously manage any of this.
Exactly. And when you consider the sheer sophistication and precision of these mechanisms we've discussed, the perfectly packaged energy bursts in ATP, the split-second refill capability from phosphocreatine, the body's ability to seamlessly switch between explosive anaerobic power and sustained aerobic efficiency depending on the need, and that elegant automatic control system driven by the simple presence of ADP, maybe the most astounding aspect is just how flawlessly this intricate energy economy runs every single moment of your life without you having to think about it at all.
It just works.
What does it tell you about the incredible resilience and design of the human body? That it can orchestrate this complex dance of molecules managing fuel supply, energy demand, waste removal, heat production, all to keep you going. It's something to perhaps contemplate the next time you feel that surge of energy or even that dip in the afternoon, connecting it back to these fundamental processes constantly at play within you.